look at graphs, more specifically line graphs. So you may ask yourself, why do we have to know about graphs in science? This is stressing me out. What is the point of this? Now, graphs are very, very important, not just in science, but math and also economics and various other subjects like so. But we need to understand what is a graph, what is the point of a graph. So a graph is a pictorial representation of data, right? And what it basically does is that it takes complex data and presents it in a simple format, right? Now, line graphs are used to do one of two things. The first thing is to display a change in a particular item over time. So if we want to observe or analyze or understand how something changes over time, a line graph is appropriate. The second reason we may use a line graph is to determine the relationship between two variables. So these variables are often referred to as our dependent variable and our independent variable. Now, the independent variable is that factor that is changed. So what is that something that we are changing? Okay, that is our independent variable. And if we want to study the impact of that change, then we would look at our dependent variable. So the line graph clearly shows the relationship between two variables, something that is changed and the impact of that change. Now, in order for graphs to make sense, we have to learn how to understand them, right? So this is what we are going to do today. We are going to understand how to read line graphs. So we have here a little experimental setup, okay? So this is an application of enzymes in a CSEC biology. So we have here a test tube, and inside of that test tube, we have a piece of potato and we have hydrogen peroxide. We see that when the hydrogen peroxide and the potato is combined, we have this gas being produced. Now, this rubber band is here. What is the purpose of this rubber band? What the rubber band does is that it traps the gas inside of this system so it doesn't escape, therefore forcing the gas to travel through this delivery tube. Okay, so when it goes through the delivery tube, it ends up in this gas syringe, and this gas syringe is going to tell us what volume of gas has been collected. Okay, so we have the hydrogen peroxide and the potato, we combine them, we have a gas being produced, the gas travels through the delivery tube and into the gas syringe. And as a result, the gas syringe is going to tell us what volume or what amount of gas is collected. Now, if we want to be more specific, we're going to look at these chemical equations below. And this chemical equation summarizes what is happening here. So let's look at the worded equation first. So we have hydrogen peroxide gives us or breaks down or decomposes to give us water and oxygen gas. And this is done with the help of this thing here called catalase. Catalase is that enzyme that we find in living things. And just a little recap, that an enzyme is that biological matter. It's mostly built from protein that speeds up the rate of a reaction without it being used up. So what the catalase does here is to facilitate this breakdown of hydrogen peroxide. Now, why is this important? Now, hydrogen peroxide can be considered a waste product of different chemical reactions occurring in the body. However, if it is allowed to accumulate in the body, in this situation, the potato, it can become toxic, right? So what the catalase does is to speed up the decomposition of this hydrogen peroxide, so it is broken down into harmless or safe products such as water and oxygen. Okay, so if we are doing an experiment like this and we want to know how does the volume or what volume of oxygen gas is produced over time, we can, of course, write this down in a table, but it's also nicely represented here as a line graph. So this is the first example. If you want to see how something changes over time, in this situation, we're looking at how much volume of oxygen gas is produced over time. We use a line graph. So this is our title here, volume of oxygen gas produced over time. And then we have our y-axis. Our y-axis is our dependent variable. This is our volume. And the independent variable is our x-axis, which is time. So as time changes, right, as time increases, as time goes on, what impact does that time have on the volume of oxygen gas produced, right? So let us see what's happening here. So if we start here at 0, okay? So we're here at 0. This represents the beginning of the reaction or the experiment. Okay? So at the beginning, we don't have any oxygen gas being produced already, okay? So after 1 minute, after 1 minute has passed, so we combine the potato with the hydrogen peroxide and we have allowed it to stand for 1 minute, we see some action. So we have about 0.5 milliliters of oxygen produced in 1 minute. And then, okay, a second minute has passed and we have just under 2 milliliters of oxygen produced right here. Okay? Then after the third minute has passed, this is number 3 here. And if we go up, we see that we have about 4. We go up and across here, we have about 4 milliliters of oxygen produced. And then at minute number 4, after 4 minutes have passed, we see that we have 6 milliliters around there. Okay? Around there. Now, what is this? What is this telling us? It tells us that as time increases, the volume of oxygen gas increases, okay? But does it stay there? No, it doesn't. So the experiment continues for 5 minutes and then 6 minutes. And then what do we observe? We observe that we don't have any significant increase in volume. So after minute 4, we see that the volume of oxygen remains constant. Okay, so we say, how do we describe that? We say as time increases, the volume of oxygen gas increases until minute 4, then it remains unchanged. Okay, so if you typically in an exam or a test or even an assignment or your labs, you're going to be asked to describe what is happening in a graph. So this is what we say. This is how we describe it. As time increases, the volume of oxygen gas increases until minute 4, and then the volume of the gas remains unchanged. So this represents the end of the reaction or the experiment. So at this point, we can deduce that all of the hydrogen gas has been used up. Okay? Sorry, not hydrogen gas, the hydrogen peroxide, sorry. At this point, we can say all of the hydrogen peroxide has been used up. Okay, so that is just an explanation of that type of graph. So we still have a little bit more to do, and we have to look at the scenarios where a line graph is used to explore relationships between two variables, the independent variable and the dependent variable. So we have our x's and our y's. We have about 6 different graphs. So once we see graphs with these shapes, we can understand what is happening. We know how to interpret. So I'm just going to make things super simple, and we're going to refer to the variables as x and y. Okay. Now, in real life, when you have your assignments, your exams, and so on, just as we saw here with this graph, we are going to, where the y was labeled volume of oxygen gas, you're going to have your labels for your y and your x-axis. So all you need to do is to just substitute x with your independent variable and your y with the dependent variable. So let's look at graph number 1. So we see that as we are moving from left to right on the x-axis, we are the independent variable, we are increasing, okay? And as we move up, we are increasing. So as we go up, our dependent variable, or our x, we see that it is increasing. So how would we describe graph number 1? We say when x increases, y increases. Okay, that is how we explain that particular shape. Okay, so for graph number 2, we can say when x, we always start with the independent variable, that is the thing that is changing. When x increases, what happens to the y? We see that the y, the numbers are decreasing. We're moving down from 4 to 3 to 2 and so on, right? So when x increases, y decreases. Okay? What's happening in graph number 3? So graph number 3 is very similar to what we just looked at previously. So we see that as x increases, as the time increases, so does your y, which is the volume of gas here. Okay, so we can write it here. When x increases, y increases until a certain point, then y remains unchanged. Okay, now let's look at graph number 4. It's a similar shape to 3, but it's just the opposite, right? So while number 3 increases, we see that y is decreasing because the numbers are getting smaller. So how would we describe this? When x increases, y decreases because we're going down until a certain point, then y remains unchanged. Okay, now graph number 5, this is fairly straightforward. We can see as x increases, y increases, but this type of shape represents an exponential curve, which means that it just continues and continues and continues. So we just say when x increases, y increases exponentially. Okay, so that continues. It just increases and increases and increases. Okay, so what is happening in 6? So as x increases, x is still increasing. It's moving from left to right. Okay, from 1 to 5. So x is still increasing. So we say as x increases, now what is happening to the y? It's not going up, it's not going down, it stays constant. So we can say that the y remains unchanged. Okay, so this is an overview of the different types of shapes that you would observe in your graphs and how we interpret them, okay, when X increases, Y would either increase or decrease, or it would remain unchanged, or etc. Okay? Now, let's look at this graph and see what's happening here. So this is a little different, but here we have two different variables, right? So we have our X and we have our Y. So what is on the X? Percentage concentration of hydrogen peroxide. So we have different experiments, right? And we're looking at the volume of oxygen gas produced. So at different concentrations or different strengths of hydrogen peroxide. So we have weaker concentrations of hydrogen peroxide and we have stronger or higher concentrations of hydrogen peroxide. And what we're looking at, if we increase or if we change, this is the independent variable, this is the thing that we're changing. If we change the concentration of the hydrogen peroxide, what effect does that have on the total volume of oxygen gas collected? So this is our total. What impact does that have on the total volume of oxygen gas collected? So let's look at this, okay? So it says as X increases, Y increases up to a certain point, and then it remains unchanged, okay? So this, just to point it out, this experiment is occurring at 30 degrees Celsius, okay? So we're going to say as the percentage of hydrogen peroxide, which is H2O2, increases, the total volume of oxygen, which is O2, increases, okay, until it gets to what? 2.5% concentration of hydrogen peroxide. And we say after which the volume of oxygen remains the same. Okay? So that is the same thing that we've been discussing before. Now, here's a little question here. It says sketch, and you may see something like this, right? You typically see these type of questions being added to a graph. And they will typically ask you to sketch the curve if the experiment is repeated at 35 degrees Celsius. So we have to ask ourselves, what impact is this going to have? Now, the shape is going to remain the same because it is this graph is going to remain the same. However, at a higher temperature, the rate of this reaction will increase. So what it means is that the volume of hydrogen gas, the total volume, is going to be produced at a faster rate, okay? So we're going to start from, because it's the same type of curve, we're going to start from the origin or 0 as well. But then we're going to do something like this. Okay, so this is a sketch, but we expect it to be very similar to the previous one. Okay, so the first one, this is our 30 degrees. This is the first one that we were looking at. And then the second one at 35. This is 35. So why is this drawn here? Why is it drawn above the original? So for temperature, we are going to expect a faster rate. So steeper slopes indicate a faster rate. Okay, now what if the experiment occurred at a lower temperature, let's say 25? Now, if it was a lower temperature, of course, we expect the same curve and everything. Then we're gonna see something like this. Okay, it's going to be a much gentler slope. Okay, so this is going to be 25. Okay, so gentler slopes indicate a slower rate. Okay, so this is just something to keep in mind. If you do chemistry, for sure, if you have a rates graph, you have a very high probability of this type of question being added. Sketch a curve to show what you would expect, right? And for biology, you may have it, right? So the question that you need to ask, this thing, for example, temperature, is it going to increase? Is it going to happen? Is this reaction going to happen faster? Or is this reaction going to happen slower? Now, if we expect the reaction to happen faster, we have a steeper slope. If we expect the reaction to happen at a slower rate, we have a gentler slope. Okay? So that is it for understanding line graphs.